This video is the last in a series of 5 videos illustrating the MICP user data tool available in CARP. Please view all parts to get a full demonstration of the tool's use and capabilities. In this video we will illustrate the multi-deviation worksheets of the tool. The multi-deviation sheets focus on deviation between predicted and measured MICP curves for large datasets and has the following features: average, graphical and numeric deviation data for large datasets. Deviation given for different capillary pressures and associated heights above freewater level. And finally, deviation given as average and absolute average. The tool consists of 4 worksheets: the current main sheet and 3 data sheets, one for each of the averaging methods. The data sheets are labeled Multidev Thomeer, Multidev Leverett and Multidev Lonoy, and they are similar in design. Data entry cells are marked by yellow background and mostly occur within the data sheets. We copy data from the deviation sheet, which is this sheet down here, and paste it into the first empty yellow column in the data sheets. Column H goes into the Multidev Thomeer sheet, and we start pasting at row number 3. Column I goes into the Multidev Leverett sheet, and column J goes into the Multidev Lonoy sheet. The sample's port type, which can be found in the graph of the deviation sheet, up here, port type 111, is entered into the Multidev Thomeer sheet in row number 2. This makes it possible to filter on port type in the results data. We repeat this for every sample in our dataset. The deviation between predicted and measured MICP curves for the complete dataset is displayed in the graphs of the multi-deviation sheet. Expected P10 and P90 deviation curves are presented for the Thomeer, Leverett and Lonoy averaging methods. The upper plot shows saturation deviation as a function of mercury capillary pressure, and the lower plot shows saturation deviation as a function of height above freewater level. The plots can be filtered by port type or all port types combined in cell B3. Note that the port type filtering list only includes port types present in the current dataset. The number of samples displayed are given in cell B8. The average and absolute average deviation for the Thomeer, Leverett and Lonoy methods are presented in cells B13 to 15 and in cells B24 to B26. The deviations are based on the selection made in cell B13. 3.